Welcome, future food safety officers. Today we embark on a journey into one of nature's most elegant and sophisticated systems, a system that has been perfecting itself for millions of years. A system that stands between life and death every single moment of our existence. We're going to explore the immune system, and more specifically, how understanding its intricate mechanisms will make you not just better food safety officers, but guardians of public health. Imagine, if you will, that your body is a fortress. But this isn't just any fortress, it's a living, breathing, constantly adapting citadel with an army so sophisticated that it makes our most advanced military systems look primitive by comparison. This army doesn't just fight. It learns, it remembers, and it evolves. It's an army that can distinguish friend from foe with precision that would make the most advanced computer envious. As food safety officers, you are the external guardians of what enters this fortress through one of its most vulnerable gates, the digestive system. Every day, you make decisions that affect millions of these internal armies. Every pathogen you prevent from entering food, every toxin you detect, every allergen you identify, you're working in harmony with the most sophisticated defense system ever created. The importance of understanding immunology and food safety cannot be overstated. When you understand how the immune system recognizes threats, you understand the very foundation of how diagnostic tests work. When you grasp the concept of antigen-antibody reactions, you're unlocking the secrets behind ELISA tests, rapid detection kits, and allergen identification systems that you'll use daily in your career. But more than that, you're understanding the human story. Every foodborne illness represents a battle between pathogen and host. Every allergic reaction is an immune system's overzealous response to what it perceives as a threat. Every successful prevention of food contamination is a victory not just for public health policy, but for millions of individual immune systems that will never have to fight that particular battle. All right, let's begin with the fundamental concept. The immune system. Imagine it as your body's highly sophisticated and incredibly efficient army, constantly on guard protecting you from an endless array of invaders like bacteria, viruses, fungi, parasites, and even abnormal cells. Without a functioning immune system, we wouldn't survive for long. Let's break down the immune system into key points. 1. Definition and primary function. Definition. The immune system is a complex network of cells, tissues, organs, and molecules that work together to protect the body from harmful substances and pathogens. Primary function. Its main role is to identify and eliminate threats, maintaining the body's internal balance and preventing disease. This process is called immune response. Think of this system as a vast intelligence network. It's not just about brute force, it's about recognition, communication, coordination, and memory. Every cell in this system is both a soldier and a spy, constantly gathering information about what belongs and what doesn't. 2. Key components of the immune system. Organs, primary lymphoid organs, bone marrow, the factory where all immune cells like B cells, T cells, macrophages, neutrophils are born from hematopoietic stem cells. Thymus, the school where T cells mature and learn to distinguish between self and non-self. The bone marrow is like a military academy and weapons factory combined. Here, stem cells, the ultimate blank slates, receive their first instructions about what kind of immune cell they will become. It's a process so precise that it makes the most advanced manufacturing systems look crude. The thymus, on the other hand, is perhaps one of the most fascinating organs in the human body. It's here that T cells undergo what can only be described as a rigorous education program. They're tested, challenged, and those that might attack the body's own tissues are eliminated. It's a harsh but necessary process. Imagine training soldiers who must be able to distinguish between civilians and enemies in the heat of battle. Secondary lymphoid organs, lymph nodes, small, bean-shaped structures scattered throughout the body acting as meeting points where immune cells gather and respond to pathogens. They filter lymph fluid. Spleen, a large organ in the abdomen, it filters blood, removes old red blood cells, and also serves as a site for immune responses against blood-borne pathogens. Tonsils and adenoids, lymphoid tissues in the throat, acting as a first line of defense against inhaled or ingested pathogens. Peyer's patches, lymphoid tissues located in the small intestine, crucial for immune responses against foodborne pathogens. Appendix. A small finger-shaped organ attached to the large intestine. Also thought to have lymphoid tissue. Cells. Leukocytes. White blood cells. Phagocytes. Cells that eat or engulf pathogens and cellular debris. Neutrophils. Most abundant WBCs, first responders to infection, short-lived. Macrophages. Larger, long-lived phagocytes present antigens to T cells, found in tissues. Dendritic cells, highly efficient antigen-presenting cells, APCs, linking innate and adaptive immunity. Let me paint you a picture of these cellular warriors. Neutrophils are like the first responders, the paramedics and firefighters of the immune system. They arrive quickly at the scene of infection, do their job with fierce efficiency, and often die in the process. Their sacrifice is so common that pus, that yellowish substance you see in infected wounds, is largely composed of dead neutrophils. Macrophages, whose name literally means big eaters, are the cleanup crew and intelligence officers rolled into one. They don't just engulf pathogens, they process them, break them down, and present pieces of them to other immune cells like showing evidence at a crime scene. They're the storytellers of the immune system, communicating what they've encountered to the rest of the immune network. Dendritic cells are perhaps the most sophisticated of all. They're like master spies who can infiltrate enemy territory, gather intelligence, and then return to headquarters to brief the entire army. They bridge the gap between the immediate non-specific response and the highly targeted specific response. And lymphocytes, key players in adaptive immunity. B cells produce antibodies. T cells, helper T cells, CD4+, coordinate immune responses. Cytotoxic T cells, CD8+, directly kill infected or cancer cells. Regulatory T cells suppress excessive immune responses. Natural killer, NK, cells, part of innate immunity, kill virus-infected cells and tumor cells without prior sensitization. Mast cells and base alphas, involved in allergic reactions and inflammation, release histamine. Eosinophils, involved in fighting parasitic infections and allergic responses. B cells are the weapons manufacturers of the immune system. When activated, they transform into plasma cells, biological factories that can produce thousands of antibodies per second. It's mass production at a cellular level that would make Henry Ford envious. T cells are the special forces. Helper T cells are like field commanders coordinating the entire immune response, calling in different types of support as needed. Cytotoxic T cells are the assassins. They identify infected or cancer cells and eliminate them with surgical precision. Regulatory T cells are the peacekeepers, ensuring that the immune response doesn't get out of hand and start attacking healthy tissue. Molecules, antibodies, immunoglobulins, proteins produced by B cells that specifically bind to antigens. We'll cover these in detail later. Cytokines, small proteins that act as messengers between immune cells, regulating intensity and duration of immune responses. For example, interleukins, interferons. Complement system, a group of plasma proteins that, when activated, work to enhance antibody and phagocytic responses and directly lyse pathogens. Cytokines are the communication network of the immune system. Imagine trying to coordinate a complex military operation without radios, phones, or any means of communication. That's what the immune system would be like without cytokines. These molecular messengers carry information about the type of threat, the location of the battle, and what kind of reinforcements are needed. The complement system is like a cascade of dominoes, but instead of falling down, each domino activates the next one in a chain reaction that ultimately leads to the destruction of pathogens. It's an ancient system, evolutionarily speaking, and its efficiency is breathtaking. How the immune system protects us, general overview. Recognition. The immune system must first distinguish between self, our own healthy cells, and non-self foreign invaders or abnormal cells, and non-self foreign invaders or abnormal cells. Response. Once a threat is recognized, the immune system mounts a targeted response to eliminate it. This can involve engulfing pathogens, producing antibodies, or directly killing infected cells. Memory, a crucial aspect of adaptive immunity. After encountering a pathogen, the immune system remembers it, leading to a faster and stronger response upon subsequent exposure. Understanding these components is crucial. Think of them as different units within an army, each with its specialized role, all working together for the common goal of defending the body. For food safety officers, knowing these basics helps in understanding disease pathology, allergic reactions, and the principles behind diagnostic kits used to detect foodborne pathogens or toxins. The concept of immunological memory is perhaps one of the most beautiful aspects of the immune system. It's the biological basis for vaccination, and it's why you typically only get certain diseases once. The immune system literally learns from experience, creating a library of threats that it can reference for the rest of your life. Now that we understand the immune system's components, let's explore the different types of immunity. Our body employs two main lines of defense, which then branch out further. These are the innate immune system and the adaptive immune system. Innate immunity. This is our body's first line of defense, present from birth. It provides immediate, non-specific protection against a wide range of pathogens. It doesn't learn or remember specific invaders. Non-specific responds to common molecular patterns found on many pathogens. Immediate, rapid response, within minutes to hours. No memory. Does not remember past infections. Innate immunity is like having a well-trained security force that can recognize general signs of trouble. They respond to molecular patterns that are common to many pathogens. It's pattern recognition at a molecular level. Skin. An impenetrable physical barrier. Mucous membranes line respiratory, digestive, and urogenital tracts, trapping pathogens. Cilia. Hair-like structures in respiratory tract that sweep pathogens out. Stomach acid. Low pH kills many ingested microbes. Lysozyme. In tears, saliva, and sweat that breaks down bacterial cell walls. Breaks down bacterial cell walls. Normal microbiota. Compete with pathogens for nutrients and attachment sites. Also produce antimicrobial substances. Your skin is more than just a covering. It's a fortress wall. But unlike static fortifications, it's alive. Constantly renewing itself. Shedding potential invaders along with dead skin cells. The mucous membranes are like sticky traps, capturing pathogens before they can establish a foothold. The normal microbiota. The trillions of beneficial bacteria that live in and on your body represent one of the most elegant defense strategies in nature. They're like having a friendly army already occupying the territory, making it difficult for hostile forces to establish themselves. Cellular defenses. Phagocytes, neutrophils, macrophages, dendritic cells. Engulf and destroy pathogens. Natural killer. NK cells. Identify and kill virus-infected cells and cancer cells. Inflammation. A localized tissue response to injury or infection characterized by redness, swelling, heat, and pain. It brings immune cells and molecules to the site. Fever. Systemic response to infection. Elevated body temperature can inhibit pathogen growth. Complement system. A cascade of proteins that enhances phagocytosis, causes cell lysis, cell lysis, cell lysis, cell lysis, cell lysis, lysis, cell lysis, cell lysis, cell lysis, cell lysis, cell lysis, cell lysis, and promotes neighboring cells. Inflammation. Interferons. Antiviral proteins produced by infected cells, warning neighboring cells. Inflammation, despite being uncomfortable, is actually a sign that your immune system is working correctly. The redness comes from increased blood flow, bringing immune cells to the battle. The swelling is caused by increased permeability of blood vessels, allowing immune cells and molecules to enter the tissue. The heat helps create an environment that's less favorable to many pathogens. The pain serves as a warning system, encouraging you to protect the injured area. Fever is a whole body response that turns your entire body into a less hospitable environment for pathogens. Many bacteria and viruses have optimal temperature ranges, and fever pushes your body temperature outside of those ranges. Adaptive immunity. This is a more sophisticated and highly specific defense system that learns to recognize and remember specific pathogens. It develops throughout our lives as we are exposed to different microbes. Specificity. Target specificity. Target specific antigens unique to a particular pathogen. Diversity. Can recognize a vast array of different antigens. Memory. Remembers previous encounters, leading to faster and stronger responses upon re-exposure. Self/non-self discrimination. Can distinguish between body's own cells and foreign invaders. Adaptive immunity is like having a detective force that not only solves crimes but also keeps detailed files on every criminal they've ever encountered. This system can generate an almost infinite variety of responses to an almost infinite variety of threats. Main branches of adaptive immunity. A. Humoral immunity. Antibody-mediated immunity. Mediated by B lymphocytes, B cells. When activated by an antigen, often with help from T cells, B cells differentiate into plasma cells, produce large quantities of specific antibodies. Memory B cells, long-lived cells that provide immunological memory. Primary role: targets extracellular pathogens, bacteria, toxins-free viruses, in body fluids, blood, lymph. Humoral immunity is like having a weapons manufacturing system that can produce custom ammunition for every type of target. When a B cell encounters its specific antigen, it's like a key fitting into a lock. This triggers the B cell to transform into a plasma cell, a biological factory dedicated to producing one specific type of antibody. B, cell-mediated immunity, CMI, mediated by T lymphocytes, CTLs or CD8 plus T cells 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 or CD8 plus T cells. Directly kill infected body cells, for example, virus-infected cells. Helper T lymphocytes, CD4 plus T cells, cancer cells, and transplanted cells. Helper T lymphocytes, CD4 plus T cells, crucial orchestrators of the immune response. They activate B cells, cytotoxic T cells, macrophages, and other immune cells by releasing cytokines. Memory T cells provide long-term immunity. Primary role: targets intracellular pathogens, viruses, some bacteria like Mycobacterium tuberculosis, and cancer cells. Cell-mediated immunity handles the threats that hide inside our own cells. Viruses, for example, are masters of disguise. They hijack our cellular machinery and hide inside our cells. But infected cells display fragments of viral proteins on their surface, like flying a flag that says, "I'm infected." Cytotoxic T cells can read these flags and eliminate the infected cells before the virus can spread. Active versus passive immunity. How immunity is acquired. Active immunity, definition, develops when the immune system is stimulated to produce its own antibodies and memory cells. It is long-lasting. Natural active immunity, acquired through natural exposure to a pathogen, for example, getting sick with chickenpox. Artificial active immunity, acquired through vaccination, for example, measles vaccine, where inactivated or attenuated pathogens or their components are introduced to stimulate an immune response without causing disease. Active immunity is like learning to fight by actually fighting. Passive immunity. Definition: acquired by receiving preformed antibodies from another source. It provides immediate but temporary protection as no memory cells are produced. Natural passive immunity. Acquired from mother to child, for example, antibodies crossing the placenta to the fetus, or through breast milk. Artificial passive immunity, acquired through injection of antibodies, for example, antitoxins for tetanus, snake antivenom. Passive immunity is like borrowing someone else's weapons. It provides immediate protection, but since you didn't learn to make the weapons yourself, the protection is temporary. A mother's antibodies protecting her newborn baby is one of nature's most beautiful examples of passive immunity. It's biological love in molecular form. Understanding these different types of immunity is vital for food safety officers. It helps you grasp how foodborne illnesses affect the body, the basis for allergic reactions, often involving IgG antibodies, and the principles behind public health interventions like vaccination and immune globulin therapy. Let me give you a practical example. When you're investigating a foodborne illness outbreak, understanding the difference between active and passive immunity helps you understand why some people in the same household might get sick while others don't. Previous exposure, vaccination status, age, and overall immune status all play roles in determining who gets sick and how severely. Now, let's zoom in on one of the most fundamental and precise interactions in immunology, the antigen-antibody reaction. This reaction is at the heart of adaptive immunity and is extensively used in diagnostic tools in food safety. Think of it as a highly specific lock and key mechanism. Definition: an antigen is any substance, typically a protein or polysaccharide, that the immune system recognizes as foreign and that can trigger an immune response. Immunogenicity, the ability to induce an immune response. Antigenicity, the ability to bind to an antibody or T-cell receptor, epitope, the specific small region on an antigen to which an antibody or T-cell receptor actually binds. An antigen can have multiple different epitopes. Think of an antigen as a criminal's face, an epitopes, and epitopes as the distinctive features: the scar above the left eyebrow, the crooked nose, the distinctive tattoo. An antibody doesn't recognize the entire face. It recognizes one specific feature. This is why one pathogen can stimulate the production of many different antibodies, each recognizing a different epitope. Exogenous antigens. Antigens that enter the body from the outside, like bacteria, viruses, pollen, food components like allergens and toxins. Endogenous antigens. Antigens produced inside the body's own cells, like viral proteins produced by infected cells or proteins from cancer cells. Autoantigens. Our body's own normal components that, under certain conditions like autoimmune diseases, are mistakenly recognized as foreign. Allergens. Antigens that cause allergic reactions in susceptible individuals. Toxoids. Inactivated toxins that retain their antigenicity but lose their toxicity, used in vaccines like tetanus toxoid. For food safety officers, understanding the different types of antigens is crucial. Food allergens are exogenous antigens that trigger inappropriate immune responses in susceptible individuals. Bacterial toxins are exogenous antigens that can cause severe illness. Understanding these categories helps you understand both the diseases you're trying to prevent and the diagnostic tools you'll use to detect them. Definition. Antibodies are Y-shaped proteins produced by plasma cells in response to a specific antigen. Their primary function is to neutralize or eliminate pathogens and toxins. Composed of 4 polypeptide chains, 2 identical heavy chains and 2 identical light chains held together by disulfide bonds. Fab region. The arms of the Y, containing the variable regions that bind specifically to antigens. Each antibody has 2 Fab regions. C region. The stem of the Y, responsible for mediating effector functions like binding to immune cells and activating complement. The Y-shaped structure of antibodies is not accidental. It's a masterpiece of molecular engineering. The 2 arms of the Y can grab onto antigens, while the stem can interact with other components of the immune system. It's like having a tool that can both identify a problem and call for help to solve it. Classes of antibodies. There are 5 main classes, each with distinct functions and locations. IgA, most abundant antibody in serum, can cross the placenta, providing passive immunity to the fetus, longest half-life. Primary antibody and secondary memory immune responses, functions, neutralization, opsonization, complement activation. IgG is the workhorse of the antibody world. It's found everywhere in the body, has a long lifespan, and provides long-term protection after vaccination or infection. When we talk about immunity lasting for years or decades, we're usually talking about IgG antibodies and the memory cells that produce them. IgM, first antibody produced in primary immune response. Pentameric structure, 5 Y-shaped units linked together, making it very efficient at agglutination. Found mainly in blood and lymph. Functions: complement activation, agglutination. IgM is like the early warning system. It's the first antibody produced when the immune system encounters a new threat. Its pentameric structure, 5 antibodies linked together, makes it incredibly effective at clumping pathogens together, making them easier targets for other immune cells. EDA. Found predominantly in mucosal secretions, saliva, tears, breast milk, mucus and respiratory and GI tracts. Usually dimeric, 2 Y-shaped units. Provides localized protection at mucosal surfaces, crucial for defense against foodborne pathogens. Functions: neutralization, prevents pathogen adherence. IgA is the guardian of the body's entry points. It's found in all the secretions that come into contact with the outside world. For food safety officers, IgA is particularly important because it's the first line of defense against foodborne pathogens in the digestive tract. e.g., low concentration in serum, primarily involved in allergic reactions, binds to mast cells and basophils, triggering histamine release. Also involved in defense against parasitic infections. IgE is the antibody that causes food allergies. In normal circumstances, it helps fight parasitic infections. But in allergic individuals, it mistakenly identifies harmless food proteins as threats and triggers potentially life-threatening reactions. ED, found mainly on the surface of naive B cells, functions primarily as a B cell receptor involved in B cell activation. EGD is like the antenna on B cells, helping them detect their specific antigens in the environment. Specificity, the most critical feature. An antibody binds only to a specific epitope on a specific antigen, much like a specific key fits only one lock. Affinity, the strength of the binding between a single antigen binding site on an antibody and a single epitope on an antigen. Avidity, the overall strength of binding between a multivalent antibody and a multivalent antigen. For example, IgM pentamer has high avidity due to multiple binding sites. Lock and key model, a widely used analogy. The antigen is the lock and the antibody is the key. The binding is non-covalent, involving hydrogen bonds, electrostatic forces, van der Waals forces, hydrophobic interactions. Types of antigen-antibody. Reactions. Effector functions. Once an antibody binds to an antigen, it doesn't directly kill the pathogen, but rather tags it for destruction or neutralizes its effects. Neutralization. Antibodies bind to toxins or viral particles, preventing them from attaching to host cells or exerting their harmful effects. Opsonization. Antibodies coat the surface of a pathogen, making it more easily recognized and engulfed by phagocytic cells. Agglutination. Antibodies bind to multiple pathogens, clumping them together. This makes it easier for phagocytes to engulf them and prevents their spread. Precipitation. Antibodies bind to soluble antigens, forming large, insoluble immune complexes that precipitate out of solution. Complement activation. Binding of antibodies to antigens on a cell surface can activate the complement system, leading to lysis of the target cell. Antibody-dependent cell-mediated cytotoxicity, ADCC. Antibodies bind to infected cells, and their Fc region is recognized by NK cells. Each of these mechanisms represents a different strategy for eliminating threats. Neutralization is like disarming a bomb. Opsonization is like putting a kick-me sign on the pathogen's back. Agglutination is like handcuffing criminals together so they can't escape. Complement activation is like calling in an airstrike. Each strategy is suited to different types of threats in different situations. Significance in food safety. The antigen-antibody reaction is the basis for many rapid and highly sensitive diagnostic tests. ELISA, enzyme-linked immunosorbent assay, used to detect specific foodborne pathogens, toxins, or allergens. It relies on antibodies linked to enzymes that produce a colored reaction. Lateral flow devices, rapid test strips. Common for on-site testing, like pregnancy tests, or rapid detection of specific pathogens. Immunofluorescence, using fluorescently tagged antibodies to visualize specific antigens in food samples or bacterial cultures. Allergen detection, crucial for identifying undeclared allergens in food products, protecting consumers with food allergies. Vaccine development. Understanding antigen-antibody reactions is fundamental to designing effective vaccines against foodborne pathogens like Salmonella or norovirus, though practical vaccines are still under development for many. Understanding food allergies. Allergic reactions are often IgE-mediated responses to food allergens, where the immune system mistakenly identifies harmless food proteins as threats. This precise interaction, the antigen-antibody reaction, is a testament to the sophistication of our immune system and a powerful tool in ensuring the safety of our food supply. Let me give you a real-world example of how this knowledge applies to your future work. When you use an ELISA test to detect Salmonella in a food sample, you're essentially using the immune system's own recognition mechanism. The test uses antibodies that specifically recognize Salmonella antigens. If Salmonella is present in the sample, the antibodies will bind to it, and through a series of chemical reactions, produce a color change that you can see. You're literally using the immune system's weapons as your diagnostic tools. Let's condense all this crucial information into 10 key points that every aspiring food safety officer should commit to memory. Immune system is the body's defense. It's a complex network of cells, organs, and molecules protecting against pathogens and abnormal cells. Innate immunity =, first, non-specific line, provides immediate general protection via barriers, phagocytes, inflammation, and fever, without memory. Adaptive immunity =, specific and memory-based, develops over time, targeting specific antigens and remembering past encounters for stronger future responses. Humoral immunity, B cells and antibodies, targets extracellular threats. Antibodies produced by plasma cells neutralize toxins and pathogens in body fluids. Cell-mediated immunity, T cells, targets intracellular threats. Cytotoxic T cells directly kill infected or cancerous cells. Antigens trigger immune responses. Antigens trigger immune responses. Any substance, usually protein or polysaccharide, that the immune system recognizes as foreign and generates antibodies against. Antibodies are specific keys, shaped proteins, IgG, IgM, IgA, 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 IgD, that bind specifically to epitopes on antigens, acting as tags or neutralizers. Antigen-antibody reaction is highly specific, a lock and key mechanism fundamental for immune protection and diagnostic applications. Key antibody functions: neutralization, opsonization, agglutination, precipitation, and complement activation are primary ways antibodies eliminate threats. Immunology is crucial for food safety diagnostics. Techniques like ELISA and rapid test strips, lateral flow, based on antigen-antibody reactions for detecting pathogens, toxins, and allergens in food. These 10 points summarize the core concepts and highlight their direct relevance to your role as a food safety officer. Make sure you understand each one thoroughly. But let me add some additional key points that are crucial for your exam preparation and professional practice. Cross-reactivity. Sometimes antibodies can bind to similar epitopes on different antigens. This can lead to false positives in diagnostic tests, which is why understanding specificity and validation is crucial. Immunocompromised populations. Understanding how immune deficiency affects disease susceptibility helps you understand why certain populations, elderly, pregnant women, immunocompromised individuals, are at higher risk for foodborne illnesses. Herd immunity. While not directly applicable to foodborne diseases, understanding this concept helps you grasp population-level disease dynamics. Exam tip. Use the mnemonic INH to remember the 4 main features of innate immunity: immediate, non-specific, always present, and has no memory. E. Summary table. Compare and contrast innate and adaptive immunity in terms of speed, specificity, memory, and main components. Professional practice tip. Always document your findings thoroughly during food safety investigations. Accurate records support public health actions and legal compliance. Now let's test your understanding with 10 quick questions. I'll read the question, give you a moment to think, and then reveal the answer. This is a great way to reinforce what we've learned. Question 1. Which type of immunity is present from birth and provides immediate present from birth and provides immediate present from birth and provides immediate non-specific protection? Answer. Innate immunity. Question 2. Which immune cells are responsible for producing antibodies? Answer. B. Lymphocytes or plasma cells, which are differentiated B cells. Question 3. What is the primary function of helper T cells CD4+? Answer. To coordinate and orchestrate immune responses by activating other immune cells through cytokine release. Question 4. Which antibody class is most abundant in blood serum? Answer. IgG, 7,580% of serum antibodies. Question 5. What is an epitope? Answer. The specific small region on an antigen to which an antibody or T cell receptor binds, also called an antigenic determinant. Question 6. Which antibody class is primarily involved in allergic reactions? Answer. ID. Question 7. What is opsonization? Answer. The process by which antibodies coat pathogens, making them more easily recognized and engulfed by phagocytic cells. Question 8. Which organ is considered the school where T cells learn to distinguish self from non-self? Answer. The thymus. Question 9. What does ELISA stand for, and what is its basic principle? Answer. Enzyme-linked immunosorbent assay. It uses antibodies linked to enzymes to detect specific antigens, producing a colored reaction when the target is present. Answer. Passive immunity. Let's continue with more challenging questions that you might encounter in your food safety officer examinations. Question 11. A food safety inspector finds that an ELISA test for salmonella shows a positive result, but bacterial culture is negative. What could be the possible explanations? Answer. Possible explanations include 1. Dead bacteria still containing detectable antigens. 2. Cross-reactivity with similar bacterial species. 3. Presence of salmonella antigens without viable bacteria. 4. False positive due to test interference. Or 5. Bacterial culture conditions may not have been optimal for salmonella growth. Question 12. Why are immunocompromised individuals at higher risk for foodborne illnesses? Answer. Immunocompromised individuals have weakened immune systems that cannot effectively recognize, respond to, or eliminate foodborne pathogens. This can result in more severe illness, longer duration of symptoms, and higher risk of complications from infections that might be mild in healthy individuals. Question 13. Explain why IgA is particularly important in preventing foodborne illnesses. Answer. IgA is the predominant antibody in mucosal secretions, including those in the gastrointestinal tract. It provides the first line of specific immune defense against foodborne pathogens by neutralizing them and preventing their adherence to intestinal epithelial cells before they can establish infection. Question 14. What is the difference between affinity and avidity in antigen-antibody reactions? Answer. Affinity refers to the strength of binding between a single antigen binding site and a single epitope. Avidity refers to the overall strength of binding between a multivalent antigen, taking into account multiple binding sites. Question 15. How does understanding complement activation help in food safety diagnostics? Answer. Complement activation can be used in diagnostic assays as an indicator of antigen-antibody complex formation. Some diagnostic tests use complement-mediated cell lysis as a readout, and understanding this system helps in interpreting results in troubleshooting assays. Question 16. What is the principle behind lateral flow rapid test strips commonly used in food safety? Answer. Lateral flow tests use antibodies immobilized on a membrane to capture specific antigens from a sample. If the target antigen is present, it binds to the antibody and produces a visible line, indicating a positive result. Question 17. Why is it important to validate diagnostic tests for cross-reactivity before using them in food safety labs? Answer. Validation ensures that the test does not produce false positives by reacting with non-target antigens. This is crucial for accurate detection of specific pathogens or allergens and for avoiding unnecessary recalls or public health scares. Question 18. What role do memory B cells play in long-term immunity, and how does this relate to booster vaccinations? Answer. Memory B cells persist after an initial infection and can rapidly produce antibodies upon re-exposure to the same antigen. Booster vaccinations stimulate these cells, ensuring continued protection against disease. Question 19. Which immune cell type is primarily responsible for presenting antigens to T cells in the lymph nodes? Answer: dendritic cells. Explanation: dendritic cells are the most efficient antigen-presenting cells, capturing antigens and migrating to lymph nodes to activate T cells. Question 20. What is the main function of the complement system in immunity? Answer: to enhance the ability of antibodies and phagocytic cells to clear microbes and damaged cells, promote inflammation, and directly lyse pathogens. Explanation: the complement system is a group of proteins that, when activated, help destroy pathogens through multiple mechanisms. Question 21. Which immunoglobulin class is most important in mucosal immunity? Answer: IgA. Explanation: IgA is the predominant antibody in mucosal areas, providing the first line of defense against pathogens entering through mucous membranes. Question 22. What is the role of regulatory T cells in the immune system? Answer: regulatory T cells suppress excessive immune responses and help maintain self-tolerance. Explanation: they prevent autoimmunity and control the intensity of immune reactions. Question 23. Which type of hypersensitivity reaction is responsible for most food allergies? Answer: type of hypersensitivity, immediate, IgD mediated. Explanation: most food allergies are caused by Antigen-Antibodies triggering mast cell degranulation and histamine release. Question 24. What is the principle behind immunofluorescence assays used in food safety labs? Answer: immunofluorescence uses antibodies labeled with fluorescent dyes to detect specific antigens in samples. Explanation: when the antibody binds its target, the fluorescence can be visualized under a special microscope, confirming the presence of the antigen. Question 25. Why is it important for food safety officers to understand the concept of herd immunity? Answer: understanding herd immunity helps food safety officers appreciate how vaccination and population immunity can prevent outbreaks, even for diseases not directly related to food. Explanation: it informs public health strategies and risk assessments. Let's examine some case studies that illustrate how immunological principles apply to real-world food safety scenarios. Case Study 1. Restaurant Outbreak Investigation. A local health department receives reports of gastrointestinal illness from patrons of a restaurant. As a food safety officer, you need to investigate. Blood samples from affected individuals show elevated IgM antibodies against Salmonella enteritidis, but however, further testing reveals that IgG antibodies are also elevated, indicating a recent ongoing infection. As a food safety officer, you must interpret these results in the context of the outbreak timeline and patient histories. Case Study 2. Allergen contamination in a food processing plant. A batch of cookies is found to contain undeclared peanut protein, leading to an allergic reaction in a consumer. Your investigation involves tracing the source of contamination, reviewing cleaning protocols, and testing samples using ELISA kits. You collect environmental swabs, test raw ingredients, and interview staff about cross-contact procedures. The ELISA test confirms the presence of peanut protein in the final product, and corrective actions are implemented to prevent future incidents. Case Study 3. Outbreak of Listeria in packaged salads. Several cases of listeriosis are reported, and traceback investigation points to a specific brand of packaged salads. As a food safety officer, you coordinate with public health authorities, collect samples, and oversee laboratory testing for Listeria monocytogenes. The investigation reveals that the contamination occurred during packaging, and the company initiates a recall. You help communicate the recall to retailers and consumers, and review the plant sanitation procedures to prevent recurrence. These case studies highlight the importance of immunological knowledge in real-world food safety practice. Understanding how to interpret antibody tests, recognize patterns of infection, and apply diagnostic tools is essential for protecting public health. Case Study 4. Norovirus outbreak at a school. Several students develop vomiting and diarrhea after a school lunch. As a food safety officer, you investigate the food preparation area, interview staff, and collect samples. Laboratory testing confirms norovirus in a salad ingredient. The outbreak is traced to a food handler who returned to work too soon after illness. Key learning. Strict exclusion policies for ill food handlers are essential to prevent viral outbreaks. Case Study 5. Botulism in home canned vegetables. A family is hospitalized with neurological symptoms after eating home canned green beans. Investigation reveals improper canning technique and lack of pressure sterilization. Laboratory tests detect botulinum toxin. Key learning. Food safety officers must educate the public on safe home canning practices and recognize symptoms of rare but severe foodborne illnesses. Case Study 6. Cross-contamination in a meat processing plant. Routine testing finds Listeria monocytogenes on equipment surfaces. Investigation shows that cleaning protocols were not followed between processing raw and cooked products. Corrective actions include staff retraining, revised cleaning schedules, and environmental monitoring. Key learning. Preventing cross-contamination is critical in food processing environments to protect public health.